Good day everyone. Today, I will discuss about early embryogenesis and material recognition of pregnancy. Before describing the important events of early embryogenesis, I will discuss several potentially confusing terms with overlapping meanings need to be defined. These terms have subtly different uses depending on the species and the context in which they are used. After syngamy, or the fusion of the male and female pronuclei, the zygote becomes an embryo. An embryo is defined as an organism in the early stages of development. In general, an embryo has not acquired an anatomical form that is readily recognizable in appearance as a member of the specific species. For example, at early stages of development, the pig embryo cannot be distinguished from the cow embryo except by skilled embryologists. As a matter of fact, at certain stages, the human embryo cannot be distinguished from the embryos of lower species. A fetus is defined as a potential offspring that is still within the uterus. The term embryo, conceptus, and fetus are often used interchangeably to describe the developing organism. A conceptus is defined as the product of conception. It includes the embryo during the early embryonic stage, the embryo and the extraembryonic membranes during the pre-implantation stage, and the fetus and placenta during the post-attachment phase. So, early embryogenesis happens when the presence of male and female pronuclei within the cytoplasm of the oocyte characterizes a developmental stage of the newly fertilized oocyte. When the male and female pronuclei can be observed, the cell is called an ootid. The ootid is one of the largest single cells in the body and is characterized by having an enormous cytoplasmic volume relative to nuclear volume. This characteristic is important since subsequent cell divisions within the confines of the zona pellucida will involve partitioning of the cytoplasm into smaller and smaller cellular units. Following fusion of the male and female pronuclei, the single-celled embryo, now called a zygote, undergoes a series of mitotic divisions called cleavage divisions. The first cleavage division generates a two-celled embryo, the cells of which are called blastomeres. Each blastomere in the two-celled embryo is about the same size and represents almost exactly one-half of the single-celled zygote. Each blastomere undergoes subsequent divisions, yielding 4, 8, and then 16 daughter cells. In the early stages of embryogenesis, each blastomere has the potential to develop and to separate healthy offspring. Identical twins are derived from the blastomeres of a two-celled embryo that divide independently to form two separate embryos. Blastomeres from the 2, 4, 8, and 16-celled embryos are totipotent. Totipotency is a term used to describe the ability of a single cell or blastomere to give rise to a complete, fully fanned individual. This illustration shows the pre-attachment development of the embryo. In the ootid, male and female pronuclei along with the first and second polar bodies are present. Fusion of the male and female pronuclei into a single diploid nucleus continues syngamy. Shortly thereafter, the zygote undergoes cleavage, or mitotic divisions, and give rise to daughter cells called blastomeres. Finally, the rapidly growing blastocyst hatches from the zona pellucida and forms a hatched blastocyst that is free-floating within the uterus. Then, cells of the morula continue to divide and a blastocyst develops. It consists of an inner cell mass and a cavity called the blastocoele and a single layer of cells called the trophoblast. Finally, the rapidly growing blastocyst hatches from the zona pellucida and forms a hatched blastocyst that is free-floating within the uterus. When a solid ball of cells is formed and individual blastomeres can no longer be counted accurately, the early embryo is called a morula. A morula is a solid mass of blastomeres resulting from a number of cleavages of a zygote, or fertilized egg. Its name derives from its resemblance to a mulberry. During the morula stage, cells begin to separate into two distinct populations, the inner and the outer cells. During this transition, there is increased expression of genes involved in cell-to-cell adhesion, molecular transport, and intra-intercell communication. This is accompanied by asymmetric division of cells that are thought to sequester differentiation factors in the outer layer and stem cell factors in the inner cell mass. Cells in the inner portion of the morula develop gap junctions that allow for intracellular communication and may enable the inner cells to remain in a defined cluster. The outer cells of the morula develop cell-to-cell adhesion known as tight junctions. These tight junctions are believed to alter the permeability of the outer cells. Tight junctions form between the outer cells of the morula, while the gap junctions form between the inner cells, thus creating two groups of cells. Sodium is pumped into the intracellular spaces by the outer cells of the morula, and water flows osmotically. Therefore, fluid begins to accumulate within the morula. As the fluid accumulates, the outer cells become flattened and a cavity known as the blastocoele is formed. The gap junctions connecting the inner cells of the morula allow these cells to polarize as a group. As a result, two separate cellular components emerge. These are the inner cell mass and the trophoblast. The inner cell mass will give rise to the body of the embryo. The trophoblastic cells will eventually give rise to the chorion, and the chorion will become the fetal component of the placenta. This table shows the timing of pre-attachment embryogenesis relative to ovulation within females of various species. Non-bolded values occurs in the oviduct, while the bolded values occurs in the uterus. This illustration shows the stages of embryonic development starting from fertilization up to implantation. Fertilization happens when a sperm cell fuses with an egg cell to form a single cell called zygote. Zygote divides into two cell stage, then four cells, and continues dividing to form an 8-cell stage. These cells keep multiplying to form a ball of cells known as morula. Next is the blastocyst formation. As the cells continue dividing, a hollow ball of cells called a blastocyst forms. The blastocyst has an inner cell mass surrounded by an outer layer of cells called the trapezoderm. There is a fluid-filled cavity called blastocoele inside the blastocyst. Next is the hatching and implantation. The hatching blastocyst breaks free from its outer shell, the zona pellucida, allowing it to implant into the lining of the uterus. Once it implants, it forms a structure called the filamentous conceptus, which elongates and continues developing into a more oval shape, or ovoid conceptus. And lastly, the migration of mesoderm. The blue cells represent the mesoderm, a layer that migrates during development to form important structures like the trilaminar trapezoderm, which will contribute to the placenta and other tissues. Development of the extraembryonic membranes. After blastocyst has been hatched, the conceptus undergoes massive degrowth. For example, in the cow at day 13, the blastocyst is about 3 mm in diameter. During the next 4 days, the cow blastocyst will become 250 mm in length and will appear as filamentous thread. By day 18 of gestation, the blastocyst occupies space in both uterine horns. Mammalian embryos can be subdivided into two primary groups. In the first group, that includes most domestic animals, the pre-attachment period within the uterus is long, which includes several weeks. During this time, extensive extraembryonic membranes form by a folding process that generates the amnion, chorion, and allantochorion. The extraembryonic membranes form after implantation or attachment. The extraembryonic membranes of the pre-attachment embryo consist of the amnion, allantoic sac, chorion, and yolk sac. Amnion is the membrane that encases the embryo in a fluid-filled cavity called amniotic cavity. Allantoic sac serves as a storage site for metabolic waste and plays a role in gas exchange. Chorion is the outermost membrane surrounding the embryo. It facilitates gas exchange and nutrient transfer between the mother and the developing embryo. And lastly, the yolk sac. Yolk sac provides initial nourishment to the embryo before the placenta is fully developed. Schematic diagram illustrating the typical development of extraembryonic membranes in mammals. The hatch blastocyst consists of the inner cell mass, the trophoblast, and the blastocoele. Very early in embryonic development, the primitive endoderm, which is the blue layer, begins to form beneath the inner cell mass and grows downward, forming a lining on the inner surface of the trophoblast. At the same time, the mesoderm, which is the red layer, begins to develop between the primitive endoderm and the embryo. Cells of the morula continue to divide, and the blastocyst develops. It consists of an inner cell mass, a cavity called the blastocoele, and a single layer of cells called the trophoblast. The mesoderm continues to grow, forming a sac that surrounds the yolk sac and pushes against the tropectoderm. The newly formed mesodermal sac pushes against the tropectoderm and begins to fold upward, forming wing-like structures called amniotic fold. The mesoderm now completely surrounds the yolk sac and the developing allantois. The allantois is a diverticulum from the primitive gut that collects embryonic waste. The mesoderm continues to fuse with the cells of the tropectoderm to form the chorion. The amniotic folds continue to grow upward around the embryo. The yolk sac begins to regress, but the allantois continues to grow and expand. The amniotic folds almost completely surrounds the embryo. The leading edges of the amniotic folds will eventually fuse. The hatch blastocyst consists of inner cell mass, the trophoblast, and the blastocoele. Very early in embryonic development, the primitive endoderm, which is the blue layer, begins to form beneath the inner cell mass and grows downward, forming a lining on the inner surface of the trophoblast. At the same time, the mesoderm, which is the red layer, begins to develop between the primitive endoderm and the embryo. And now, I will be discussing maternal recognition in different species of animal. The critical series of events by which the conceptus initially signals its presence to the dam and enables pregnancy to continue is referred to as maternal recognition of pregnancy. In most species, the conceptus must provide a timely biochemical signal or the pregnancy will terminate. In order for the events of early embryogenesis to continue into an established pregnancy, luteolysis must be prevented. Progesterone must be maintained at sufficient high levels so that embryogenesis and attachment of the developing conceptus to the endometrium can take place. The embryo enters the uterus between days 2 and 5 after ovulation. Corpus luteum of ruminants produces oxytocin that stimulates endometrial cells to synthesize PGF2 alpha. The production of PGF2 alpha is dependent upon the threshold number of oxytocin receptors that are synthesized by endometrial cells at a critical time during the estrous cycle. When these receptors are available in sufficient numbers, pulsatile secretion of PGF2 alpha occurs in response to luteal oxytocin secretion and luteolysis follows. Clearly, this mechanism must be prevented if a successful pregnancy is to proceed. This illustration shows a comparison between the endocrine condition of the female with no conceptus present and with conceptus present. Notice that in the pregnant animal, episodes of PGF2 alpha that cause luteolysis do not occur. These are blocked because endometrial oxytocin receptor synthesis is blocked. This is called maternal recognition. Maternal recognition must occur prior to the onset of luteolysis if the pregnancy is to be maintained. Ewe and cow, the blastocyst secretes materials that block the synthesis of uterine oxytocin receptors. Interferon 1 is secreted by the trophoblastic cells of the blastocyst. Interferon 1 acts on the endometrial cells of the uterus to inhibit the production of oxytocin receptors so that oxytocin cannot stimulate PGF2 alpha synthesis. In addition, interferon 1 causes secretion of proteins from the uterine glands. The arrows from the uterine glands indicate the movement of products that are secreted into the uterine lumen to nourish the conceptus. Finally, interferon 1 can leave the uterus via the uterine vein to affect the ovary and circulating immune cells. In the sow, two major differences exist in the maternal recognition of pregnancy compared to the ewe and cow. First, the conceptus of the pig produces estradiol that serves as the signal for maternal recognition of pregnancy. Second, PGF2-alpha is produced in significant quantities but is rerouted into the uterine lumen. The conceptus begins to secrete estradiol between days 11 and 12 after ovulation. The production of estrogen does not inhibit the production of PGF2-alpha but causes the PGF2-alpha to be secreted in a different direction than in the cycling sow. This table shows pregnancy recognition factors, critical days of pregnancy recognition, and time of conceptus attachment in mammals. The equine conceptus must make extensive contact with the endometrial surface to initiate and complete maternal recognition of pregnancy. In the mare, the presence of the conceptus prevents luteolysis. Also, in the presence of the conceptus, endometrial production of the PGF2-alpha is significantly reduced. A unique feature of maternal recognition of pregnancy in the mare is that the conceptus is translocated over the endometrial surface by uterine contractions. The conceptus is moved from one uterine horn to the other. This movement must occur between 12 and 14 times per day during days 12, 13, and 14 of pregnancy in order to inhibit PGF2-alpha. The intrauterine movement of the equine conceptus appears necessary because the conceptus does not elongate as in other species. Therefore, there is less contact between the conceptus and the endometrial surface. In other words, the movement of the conceptus is probably necessary to distribute pregnancy recognition factors to the endometrial cells. Like other species, the conceptus of the horse produces proteins that apparently have some effect on the recognition of pregnancy. The first picture shows the transuterine migration of the equine conceptus. Each black spear represents a stopping spot in which the conceptus will spend between 5 and 20 minutes. The migration of the conceptus probably distributes pregnancy factors over a wide surface of the endometrium. The second picture shows the uterus of a mare at day 14 of pregnancy. The uterus has been incised on the dorsal surface to expose the spherical conceptus. This specimen shows the conceptus and uterus on the last day, which is the day 14 of the uterine migration phenomenon. In the woman, maternal recognition of pregnancy is provided by a hormone called human chorionic gonadotropin. At about the time of implantation, at day 7 to 9 after ovulation, the human concepts begin to secrete a hormone called human chorionic gonadotropin. This is an LH-like hormone that acts on the corpus luteum to inhibit intraovarian luteolysis. The precise mechanism whereby the HCG blocks luteolysis is not known. Regardless, the luteotropic effect of HGC is sufficient to allow for implantation and maintenance of pregnancy. Maternal recognition of pregnancy in the dog and in the cat probably does not require a signal from the conceptus. In the bitch, the CL of pregnancy and the CL of the cycle have similar lifespans. Therefore, under normal cyclic conditions, the CL is long-lived. When luteolysis does occur, it is near the end of the normal gestation period. In other words, the period of diestrus is quite similar to the gestation period, and thus the corpus luteum is not lysed under normal conditions until the gestation period is complete. In the queen, which is an induced ovulator, if mating does not occur, corporeal lutea are not formed, and a post-estrus period of several days, which is 8 to 10 days, exists before another estrus. In the queen that has been bred, a CL forms and the duration is the same as gestation, which is about 60 days. Like the bitch, a signal from the conceptus is not needed because corporeal lutea are not lysed before a pregnancy is established. A successful pregnancy requires maintenance of high blood progesterone concentrations. Regardless of whether or not specific pregnancy recognition signals are provided, progesterone concentrations in the blood of the dam must be maintained at a sufficiently high concentrations so that the conceptus will grow and develop. The extraembryonic membranes will form an attachment with the endometrium to provide a semi-permanent link between the dam and the fetus.